Hello everybody, I am R.S.AKSHAYA here to present a video presentation on the topic Chemiosmotic hypothesis. Let's continue further to explore more. So let's discuss the mechanism of chemiosmotic hypothesis. So for any process to occur, there will be some basic requirements. Here, for the chemiosmotic hypothesis to hold good for or to apply to that condition, it requires few basic components, or you can call them to be prerequisite components for this phenomena to occur. So those basic components are proton or electrochemical gradient. It can be called as the proton gradient or the electrochemical gradient that acts as a main driving force for this theory to be applicable, and then a proton pump. If there is a gradient, we require a pump to regulate that gradient. So necessarily we require a proton pump, and we also require ATP synthase enzyme. Why? Because we need to produce ATP. So that process of ATP synthesis is governed by, or it is catalyzed by, ATPase enzyme. Thereby, it is also important for this process to completely occur. And then we also require a membrane such as the thylakoid membrane that allows the movement of electrons and protons across it. So here this membrane is permeable to all these movement of electrons as well as protons across it. So here we are explicitly dealing with the phenomena of chemiosmotic hypothesis with respect to photosynthesis. So here we are considering such a membrane which we require to be the thylakoid membrane. So these are the basic requirements or the prerequisite constituents for this theory to be applicable. Consecutively, let us discuss the mechanism of chemiosmotic hypothesis. Here we have a thick bordered pink colored ellipse-like shape that represents thylakoid membrane. This is the outer surface of the thylakoid membrane, which is filled by stroma, and this is the inner surface which is covered by lumen. And here the process begins with the photolysis of water. This special center is called the oxygen evolution center, where photolysis of water occurs in presence of sunlight to produce oxygen and hydrogen ions and some electrons. In the meantime, the PS2 is irradiated with light, because of which it becomes excited and hence it extrudes electron. Now there is a vacancy of electrons in PS2 which is filled by the electrons produced as a result of photolysis. Further, as PS2 passes electrons to the proton carrier plastoquinone transports 2H plus for each and every electron it carries from stroma to lumen, and then the electrons travel through cytochrome, B6F, plastocyanin, and then PS1, ferredoxin and protein, and then as a result of action of NADP reductase, NADPH is formed from NADP plus. And then here, as we can see, lumen has higher concentration of H plus ions comparatively, which is more than the concentration of H plus ions in stroma. Thereby, naturally, H plus ions move from the lumen to the stroma. So, whose channel is guarded by and whose path is guarded by the CF0 and F1 particle or CF1 particle. Now, this is the tail piece and this is the head piece. Now, the tail piece CF0 helps in the facilitated diffusion of protons, and then for the protons pass through CF1, which creates conformational changes due to the energy release in CF1, and thereby the active sites of the ATP synthase in CF1 gets exposed. because of which the substrates here, which are ADP and P inorganic, that is inorganic phosphate, gets attached to ATPase in order to produce ATP. Here we have a flow chart for this hypothesis. Firstly, irradiation of PS2 occurs. Secondly, electrons are extruded from pheophytin. Here pheophytin is nothing other than it is the electron acceptor of PS2. Hence electrons are extruded from pheophytin, and then photolysis of water occurs in oxygen evolution center. Another point is that as the electrons are extruded from pheophytin, here we can generally say it as PS2, there is a vacancy created for the electrons, and those electrons which have been given from pheophytin passed into the ETS, now it has to be compensated, and it will be compensated by the electrons which were released as the products of photolysis. Now here the third step is electrons are transferred into PQ and then into cytochrome B6F and subsequently into plastocyanin, and this represents the ETS. Further, electrons are transferred to PS1, and then we have some protein complexes through which electrons travel, and finally H plus gradient is generated. Now H plus gradient is generated, which will help in the synthesis of ATP. Movement of H plus from lumen to stroma, and then F1 and F0 complex come into action now, and finally there is a conformational change. Conformational changes are produced which results in the ATPase activity, thereby ATP is synthesized. Consecutively, let us see some important facts to remember with respect to chemiosmotic hypothesis. So the first one is that 4 electrons will be furnished by the photolysis of one water molecule. Now the second one is 2 water molecules will be involved in photolysis to produce one oxygen molecule. And then we have another fact, for one electron which is transferred via PQ, that is plastoquinone, 2H plus are transported from stroma to lumen. And then for transport of one electron, 4 photons or light particles, which can also be called as quanta, are required. Total number of H plus transferred and which get accumulated in the lumen are 12. And how do we get the number 12? 8 from the ETS, means for one electron transport 2H plus is pulled or accumulated into the lumen. That's the point how we get 8, and 4H plus from photolysis of one water molecule. And then for synthesis of one ATP, 4H plus are required, and for the synthesis of one NADPH, 2 electrons are required. Another point which we have to discuss is that these electrons which are produced by photolysis will be helping in compensation of the vacancy of electrons due to electron extrusion from PS2 when it is excited after the irradiation of light. Thereby, we come to the conclusion of this topic. I hope this module helps you in understanding the concept of chemiosmotic hypothesis. Thank you for watching.